Hey everyone. So this is going to be the first of two lectures that I'm going to have for you on control of gene expression. So we're going to take this particular lecture to talk about how prokaryotic cells do this, and then the other lecture will have eukaryotic cells. So let's go review what's happening when we talk about gene expression. So this is nothing new, right? So we talked about this before. We have our genetic material stored here in the DNA. We then transcribe that into messenger RNA, and then that gets translated by the ribosome into protein. So when we do this DNA to RNA to protein, we have these two processes, transcription and translation, and we can actually control gene expression at either one of these levels. So when we go from DNA to RNA, we can either block or enhance that process. When we go from RNA to protein, we can block that process at the ribosome. And then in eukaryotic cells, as we'll see, some eukaryotic cells have some strategies here to deal with proteins. And so we'll talk a little bit about that in the second part of this. So we're going to talk about how prokaryotes do this. And prokaryotes do this mostly at this DNA to RNA level. So they're controlling gene expression at a transcriptional level. So let's use an example to think about this. Say you guys were to eat donuts. Now I know this is a little silly. We talked about donuts being brain food because of all the glucose in the donuts. But let's say, sorry about that, let's say that we have, you guys get some donuts, you drink some milk, that's going to go in your digestive tract. In your digestive tract, you have a whole bunch of these bacteria called E. coli. They're the main bacteria that live in your gut. And they eat whatever you eat. And they help take care of you. They help maintain a healthy gut. They make some vitamins for you. But they're going to eat what you eat. Now, E. coli would love just to eat glucose because that's a really easy fuel to use. In fact, their genes are set up so that when they get glucose, they can easily metabolize it. But if you give them milk, they can actually still live off the lactose, that milk sugar that you have in glucose. And remember that lactose is two sugars, right? So lactose is the sugar glucose, and it's the sugar galactose. Those two things are stuck together. So E. coli have to be able to take the lactose. They have to break that disaccharide into two monosaccharides. And then they have to be able to convert that galactose into glucose. And so they need the genes to do that. Now, they don't eat milk all that much. So they don't need to actually do this until they have milk present. And so we're going to look at this thing called the Lac Operon. So the Lac stands for lactose. The Operon actually stands for how bacteria work their genes. And we understand this pretty well because we've been studying bacteria for a while. So let me show you what this Lac Operon looks like. Now, the Lac Operon is a little weird. The Lac Operon actually consists of two things here. It consists of this region, and let me get my pen going here. It consists of this region right here. This region is called the regulatory region. And then the other piece of this is this piece over here. This piece over here is my coding region. And so the operon is actually these two pieces. Now, the operon in bacteria works this way. You'll notice there's three genes. This one's labeled LAC-Z, this one's LAC-Y, and this is LAC-A. These three genes are used by the cell to basically break lactose into glucose and galactose, and then modify it into the galactose part, into glucose. So all the genes we need to process that are right here. And bacterial cells is the way it works. The genes are all stacked together, so when we express one, we express all of them. Now, this part is the control region, and there's a lot of stuff going on here. So there's this promoter, there's this repressor thing, there's a cap binding site, there's a promoter. Now we've talked about these promoters before. This is the important one. This is where the RNA polymerase is going to bind to the DNA molecule. So I'm going to stick RNA polymerase right here on this promoter, and when I do that, if nothing's in the way, I'm going to turn these genes into messenger RNA. Now the important piece here is this other piece called the operator, because the operator is going to be the on-off switch for this entire set of genes. So an operon is basically a control region with a promoter and an operator, and then a gene region that has all the genes we need for a particular thing that we're going to do. So let's turn that off, and then let's look here at our next thing. So here's how the Lac Operon works. And there's kind of a lot going on in this diagram, so bear with me. So this is showing you here is my control region, right? So there's actually a gene here called the Lac repressor gene that codes for this protein, which is pink. That is actually my repressor. That turns off the gene. There's a binding site here for this thing called CAP, and that's a protein that helps regulate expression of this gene. Now, we talked about this before, CAMP, cyclic AMP, that's a cell signal. This is produced in the presence or absence of glucose. We're not going to get into a huge level of detail on control, but that's what that's here for. And then down here in green, this is my RNA polymerase, and it's bound to the promoter. So that's going to happen no matter what. Now, this gene is currently in the off position. So on the operator, we've stuck here this protein that we built over here in the repressor region. So this just gets turned on. So this protein is always there. This is called the Lac repressor gene, and it sticks to the operator whenever there's not milk present. So when the RNA polymerase goes to read the DNA to make messenger RNA, it can't. It gets blocked by this repressor. So in this case, my operator is acting like an off switch. Now, this is what we call positive control, because if I sprinkle milk on this thing, and that's what they're showing you down here. Let me scroll down just a second here. If I add milk, so these yellow dots represent a form of lactose that actually acts as an inducer. It sticks to this protein and changes the shape of this protein so that it doesn't bind to the operator anymore. And that means my RNA polymerase can go express those genes. And as soon as that happens, you can see here we're making messenger RNA. These little orange red things are my ribosomes. They start making the protein. So they're going to translate proteins. These three proteins are going to be used to break down that milk sugar. So in this case, I have this really nice, elegant little machine. It only turns on when I have milk present. If milk isn't present, this thing isn't get turned on, and that's great. I don't want to express these genes unless I actually have to break down milk. So this is the operon, where we have a control region that includes a promoter and an operator, and then a gene region. And in this case, for the Lac Operon, when you have a protein present, in this case lactose, that binds to this repressor protein, once that happens, we express the genes. But as soon as you take away that lactose, the protein goes back to its proper shape, it sticks to the DNA, and we turn off the gene. So it's a nice little elegant machine for regulating gene expression.